Okay, so my name's Jonathan Cohen. I'm going to talk to you about some diagnostic methods of assessing whether you need nucleotides, and also about diagnostic methods in general regarding blood chemistry analysis, which is an area of speciality of mine, and illustrating health functionality and conditional need for nucleotides. I should say that I think it's about 3 years ago now, I happened to walk past a stand at Chem Expo and I saw nucleotides, and at that time neither Rachel or Peter were there, but Dean, their designer, was, and I kept asking him questions about them, and he threatened to hit me unless I came back the next day. But I stood there and I looked at it and went, who knew? I mean, I thought I knew something about nutrition and I knew nothing about it. At least that's what I thought, nucleotides. So the next day they turned up, and I haven't looked back since, because I've been using nucleotides ever since, and colleagues that know me know that all I keep saying to them is nucleotides rock, and I'll say to all of you, nucleotides rock. And about a year ago I was at a CAM Expo, or CAM conference, and Rob Kirk was there doing a keynote speech, and he said, I seem to recall one of the 3 key things he was talking about, one of them was nucleotides, and I thought, there's a man that knows. And of course he does know, and he's written some very good articles on nucleotides, and you should all know as well, and if you're not using them, I insist that you do, because not that I have any stake in this company, unless Rachel wants to give it to me, but you will get results across the board, and that's a hell of a statement for any clinician to make, but that's my experience. I know Gunva will be coming up and she'll be mentioning something similar. There are other people here that I know have got experience of nucleotides, and they'll concur with me. So I'll crack on and just talk a bit about blood chemistry, and then I'm going to go on to blood chemistry and assessing the need for nucleotides, and then I'm going to be shameless in selling my blood chemistry software to you, or analysis and reports. So blood chemistry is essential. I mean, everybody knows they've got blood. It's red. It leaks when you get injured. We use functional ranges versus pathological ranges, which is really important. So we're not just looking at the ranges that you will get from your doctor or your medical plan. We're looking at old-time ranges where once upon a time were considered healthy. Blood shows homeostasis. So the blood that's at the tip of your toe and the blood that is right at the top of your head has got to be the same. The body doesn't differentiate, and you don't suddenly get a different drop of blood in different parts of your body. Every cell has got the right to the same amount of blood. It's equality for you. It's sophisticated. As practitioners, it increases the scope of work, because you can actually give people health roadmaps, and it identifies the need for and use of nucleotides as an essential part of practice. And get better results. It's for health professionals only. Very briefly, this is the team. The whole thing was put together by Dick and Weatherby some years ago. A couple of years ago, he said, I've managed to take what you used to do by hand, which used to take me hours and hours and hours, and I put it into a software program. Are you interested in beta testing it? It took me about a second to say yes, and haven't looked back, and we've since now formed an alliance where we brought this to the UK and Europe. Backed by Justin Price of Regeneris Laboratories for the logistical side of getting your blood drawn. He deals with all that kind of thing, and then there's me, best to last, I put it. And this is who I am. I've got 25 years of clinical experience. I specialise and like to specialise in complex cases. I don't know why, because it means I don't get to bed very much, because I'm always trying to work out what's going on. But I'm fascinated by Lyme and ME and cancer and chemical sensitivities. I'm also a Nutrigenetic Counsellor, and I do blood chemistry as a matter of rote with my clients, and I try and insist that they have their genetic makeup done as well, because I like to know what I'm giving them is appropriate and right, and it suits their particular makeup. I also do some nutritional advice, and I'm a teacher, at times a mentor and consultant as well. So blood chemistry analysis is functional testing. I mean, it should be the very first test carried out in all patients. I'm extraordinarily biased, but this is 9 years now of working with it, and it works incredibly well. I see things long before it ever gets to a pathological state. I can see when people are heading over the cliff and try and stop it. From a therapist's point of view, this covers wider breadth and knowledge than of any other test out there. We can see nutritional status, albeit functional. We can see health trends. Getting patient compliance isn't really a problem because the test that patients usually have as the first rule of thumb is blood test. You go to a doctor, you get a blood test. It's a normal, and people understand this. So if you're going to be assessing them and you're going to say, well, I think you ought to be on nucleotides, you can show them from the tests, and I'm going to show you as we go through exactly why. And even if they can't and they don't want to get a blood test drawn, they've got access to one they've done previously. So it's always possible to get an assessment at any time of what they've been going through, how they've been going through, and also look across a series of tests and see what the development of any problems may be. 60 to 70% of all decisions regarding patients' diagnosis, treatment, etc. are based on laboratory tests. So laboratory test results are important. People understand them. Irrespective whether you're practitioners or whether it's clinical diagnosis, it's an essential part of making a clinical diagnosis. Even though as a CAM practitioner I'm not allowed to or not supposed to make a clinical diagnosis, I can make an assessment for health trends. So improves decision making, reduces uncertainty. I've got objective data, and I can confirm what I'm thinking when I've looked at somebody's health or status, or I've looked at their state of their tongue or their nails or their eyes, or I've listened to them. It can confirm my thinking on that or give me an additional pathway to consider. And of course I can assess the efficacy of the treatment that I'm providing for them, and I can assess their progress. And the problem that we have, or I would say that doctors have in general, is that the results which are usually provided, and most tests are provided, they're provided in a linear fashion. I don't understand why this is the case. It's a bane of my life, frankly, and I see this with whether it's organic acid testing or anything else. I just see a long list of data, and then I've got to try and take all that data and put it together into an algorithmic format. So, I've said, does one swallow a summer make? If you see an elevated marker, well, does that mean that you've got a problem, unless that marker is specifically related to a problem? In other words, does elevated thyroid-stimulating hormone mean there's hypothyroidism? You need to take other markers. So there's loads of possibilities for markers out of range. So couple them together with algorithms and give meaning to all the ranges that are there. So we use these algorithms to establish health trends, and medical algorithms are essential. It's essential that they can be computerized, automated. It's efficient, and it stops having loads of other tests having to be ordered, saves people money and time. We're not diagnostic. I'll repeat this, and I keep saying it all the time. Diagnosis is the province of doctors and the medical profession. What I do is identify health trends, and I try and do it in a low-cost and affordable way. And I want to make sense out of linear reporting. It's time efficient, it's productive, and allows a system-wide assessment. So if we look at what a typical NHS report is, a one-page report, there's little to comment on. Imagine you get extensive ones, 2 or 3 pages in some cases, but this one's got 27 biomarkers. It's in black and white. It doesn't really show you other than mean corpuscular hemoglobin is elevated and sodium is slightly elevated, and that's all it says. And if a clinician looks at that, it's going to look, well, there's not enough really to go on. You're all right, go away. But how many people go to their doctors and they hear the same thing? Nothing there. You're okay. Go away. Or you need to be on SSRIs because you're clearly depressed. That is the way of the world. It's an easy get-out clause. However, here's another one from a very well-known lab in London. 60 biomarkers, 3 pages. it's easier to read because anything that's out of range, whether it's high or low, is in red. So your eyes automatically drawn to that. But then you're drawn to it, and your eyes are drawn away from whatever else is going on because you're looking at these ranges here, which are clinical or pathological ranges. Well, when you fall outside of those ranges, you're pathologically ill. There is something wrong with your pathology. Now you have a problem. And now that problem is going to exist in dealing with it, usually from a medical perspective, which means a pharmaceutical. Pharmaceutical is likely to create side effects. Side effects means another drug at some point to counteract the side effects. And so the vicious cycle begins, whereas you just can't see health trends, let alone the need for nucleotides. The assessment of health and nucleotide needs. So from reporting, when we get a blood test report, it looks something like this. This is one of the report snapshot of it. We can see red being above range, yellow being low range, above optimal, functional, above standard or clinical. And when you see a little triangle like that ratio between sodium and potassium, means you really want to have a good look and see what's going on. And if necessary, refer on into primary healthcare providers. We know that the absorption of nucleotides, we only get 5 to 15% at best. And some of these slides you will have seen, and I've freely borrowed them from Peter and Rachel, and I had a couple of slides from Great Plains coming up. We know that in order for nucleotides to be absorbed, they need an enzyme, which is an alkaline phosphatase esterase enzyme. Like alkaline phosphatase as an enzyme, this is a zinc-dependent enzyme. Zinc dependency or zinc problems are prevalent. The nucleic acids in the cells are well protected, so getting the nucleotides out is really difficult at the best of times, so you need this enzyme. And if it's low, then you have a problem. In this case here, we're looking at alkaline phosphatase of 36 international units per litre. 36, a functional range of 70 to 100. It's not reported as being out of range because the clinical range is 35 to 115. So you haven't got a problem, but now you need nucleotides. So if you have this range of 36, you want to, as clinicians, you want to consider low zinc status. Okay. Consider poor alkaline phosphatase esterase function. In other words, you're not breaking the nucleotides out. So dietary, you may be eating the right kind of foods. You may be, getting kidneys and bone marrow and everything else in there, but you may not be getting the materials out. You might want to consider cryptopylurea, for example. I mean, that's a state of, low zinc. Best absorption, anyway, as we've said, is 5 to 15%. So if zinc is low, make an assumption that there's poor intake from food. This means you're going to have to get your nucleotides from the de novo or the salvage pathways. And we're going to come on to that and see why they may be compromised at the same time. You want to consider what is the patient diet. Factor that in as well. And consider that if the intake is poor, then the body relies on the salvage pathways. It relies on the de novo synthesis pathways. These can be very energy dependent. If you've got somebody who is energy poor, then their abilities to divert energy in those areas is going to be compromised. So we know that from dietary sources, because we'll look at the diets, and animal products have a load more nucleotides in them than plant-based sources. It doesn't mean to say that suddenly all vegetarians or vegans have to switch over to being carnivores, but in the cases of vegetarians or vegans or whatever, when their systems go out and their immune systems get compromised, they have to find other sources. This is a great way of maintaining them in their status or their preferred method of eating. The purines are widely available. Purines, everybody can get hold of purines from food. May not get it out of the food very well, but they certainly can get it because purines are principally in the energy-producing components, and everything that's alive today is creating ATP, and we can get purines from there. Pyrimidines, on the other hand, that's the cytosine and thymine. That's another problem. And if you try and research pyrimidines and you try and look for the studies and the issue there, it's really complex. There's not very much out there, and that's one of the things that makes the nucleotide nutrition organisation and the products incredible because of the pyrimidine content. Liver is one of the prime sources, offal, in bone marrow. One of the reasons why the GAPS diets can be so effective is because the amount of nucleotides that are being produced. I always remember that I was told growing up that chicken soup was Jewish penicillin. Now I understand why. Because you take the carcass, you boil it to death, you get all the nucleotides out, you've got lots of collagen, and here you go, collagen plus nucleotides are really going to affect the gut in a very, very positive way. Eggs. Broccoli's got quite a decent amount of pyrimidines, as soya's got a bit, and cauliflower as well. But does your client eat liver, offal, bone marrow? And are they zinc challenged? And if they are, then you need to consider looking at other factors. We saw this slide earlier. This is a comparison between, this is the meat world and this is the vegetarian world. And I've drawn a line pretty much at the same level. And I think you can see from the blue levels there that the amount of pyrimidines available in the vegetarian world is quite limited. You have to eat a lot of vegetables to get your pyrimidines. So let's, when we look at our reports, we look at our digestive markers, and poor digestion impacts on bioavailability of nucleotides. So let's consider the needs here. So looking at the GI system, the algorithms that we use, we will look at protein, urea, phosphorus, creatinine, globulin, eosinophils. All of those will go together to create an algorithm. If I was looking at hydrochloric acid, I'll be taking all of those and adding to it MCV, mean corpuscular volume, mean corpuscular hemoglobin, calcium, phosphate or phosphorus, iron, chloride, alkaline phosphatase, and I'd look at the anion gap, which is a seriously important marker and rarely used in the medical world, but quite simply the difference between the cations and anions, the positive ions and the negative ions, giving you a result which can be interpreted as either acidity or alkalinity. Usually in this day and age and the dietary habits that we have, I see it as mainly people as acidity, as acid. Often it's pretty high, so we're looking at a systemic acid situation that needs to be overcome. So alkalizing the diet becomes really important. Pancreatic functions, gallbladder, for example, GGTP, total bilirubin, cholesterol, alkaline phosphatase, triglycerides, direct bilirubin, and lactate dehydrogenase, and of course we've got vitamins as well. All of these factors need to be brought into play. And when we want to see the effects, which you've seen already, we can see that the effects of nucleotides on the gut mucosa is seriously incredible. The fact that within 3 weeks, damaged villi are growing back to this. The effects, because we live in a world where so many people now tend to be gluten challenged, the possibilities for people who have villi damage, whether it's through celiac or autoimmune conditions and others, is quite staggering. And a healthy gut is a healthy immune system, is a healthy life. We've heard about the effects on the bifidobacter. Having a healthy gut flora is going to lead to healthy living as well. So when we move on to other sources, when we look on salvage and recycling, the salvage pathway requires energy. It's an enzymatic pathway. The problem here is going to be whether the enzymes are being produced, the effects on through the genetic propensity, the folate pathways. It's only 70-80% efficient at best. So you're already losing up to 30% efficiency at maximum. And we want to consider the patient's energy status. So how are they expressing their energy levels? The thyroid, most tests from your GP will consider TSH and free T4, and that will be it. And on that single marker, or two markers, because TSH isn't really a thyroid marker, it assesses the pituitary gland, a clinical decision will be made that your thyroid is functioning perfectly okay. I see hypothyroidism or subclinical hypothyroidism in an enormous amount of people, usually women, sadly. And those markers not good enough. You really do need TSH, free T3, free T4, and total T3. You want to see the total of the bound and unbound thyroid hormones. So when we get a report back and we've tested it, we'll get a report that says, well, there's immune insufficiency. Well, we know if there's immune insufficiency, we know that there's going to be a need for nucleotides because the pyrimidines are essential in the production of immune functioning, and you need to make immune cells their proteins. Lack of pyrimidines means that there is a lack of appropriate RNA being made. Lack of appropriate RNA means that you are not going to be making enough protein. If there's a need for B12 or folate, you need to supplement with B12 and folate. Again, you're going to think about nucleotides to overcome these things because you're going to circumvent pathways. I think shortcuts are fantastic. Now, the thing to remember, of course, about nucleotides is that they are not supplements. They're food. They exist in food. They're in all the foods. The question is whether you can get at them or not. They are evolutionary 2 stages up from the very beginning. I mean, in the beginning, there was a big bang, and then there was some amino acids, and the next thing you know is you've got nucleotides, and then you get DNA and RNA. So, you're going back to basics. I mean, that's beautiful, in my opinion. And, of course, if we're looking at thyroid, what do we look up for thyroid? And we get a full report here of exactly what's going on, what the rationale was for that, what elements were considered, and what results weren't available, and what percentage do we think, algorithmically, that this condition actually has a validity to be considered. If we come on to the de novo synthesis, that's actually creating nucleotides from new. We have to be wary that there are certain things which are going to affect that, like infection and stress, which you've heard today. So let's just look here briefly at the way that uracil is produced. So this is going through the folate pathway. I'm just going to mark over here between dihydroorotate and orotate, which takes us all the way down through to thymine, which then takes us down to uracil. I've got a little formula here which says U plus CH3 equals T. So take uracil, methylate it, and you get thymine. And this is really critically important because uracil you find in RNA. RNA ultimately comes back, it's got to become DNA again, but you can't put uracil into DNA, except in the case where you can't methylate it. And if you do, uracil slots into where thymine should be, and the next thing you know is you've got dysfunctional DNA. Now, I hypothesize that this dysfunctional DNA with the uracil in it may be a strong link between that and cancer because of the links between folate levels and cancer. And I'll show an organic acid marker for thymine because you can actually measure uracil and thymine in the organic acid test. And highly elevated amounts of thymine can be linked to cancer. And it might be that the high elevation may be down to the fact that the uracil is not being methylated. It's just a hypothesis, but worth considering. As I mentioned, stress and disease or immune system can have a huge effect. For stress, sodium, potassium, the sodium-potassium ratio, glucose, insulin, eosinophils, chloride, triglycerides, DHEA, other markers for pathological dysfunction and inflammation markers, which I'm not going to list them out, all of those will go into assessing. And all this assessment just gives you greater weight for realizing that nucleotides rock. They should be included in everything, every day, by everyone, everywhere. And then if you have got stress, an adrenal insufficiency profile from the markers that were done because potassium was up, the sodium-potassium ratio was down, chloride was down, cholesterol, total cholesterol was elevated. It could be DHEA was depressed as well. This will give you an indication that there is stress going on. If there is stress going on, overcome stress by putting in nucleotides because you get a better recovery. Most nucleotide synthesis occurs in the liver. So if your liver is dysfunctional, as you've heard, your nucleotide availability is going to be reduced. So in which case, if we're looking at liver cell damage and if you're looking at liver dysfunction, anyone or all of these markers from ALT, LDH, AST, bilirubin, etc., all the way through cholesterol, can be put together to create an algorithm to assess liver function. Fatty liver is becoming more and more prevalent as obesity levels and sugar dysglycemia across an entire nation rises. So fat gets deposited in the liver. Now you've got a dysfunctional liver. If you have a dysfunctional liver, you're going to have a dysfunctional ability for nucleotides. If you've got a dysfunctional ability for nucleotides, and so the vicious cycle goes on. Nucleotides are conditionally essential. And again, this is just a snapshot from somebody's blood test, and we can see here that GGT is elevated. We can see that their liver markers are starting to become elevated. AST is clinically elevated. This is not healthy. This is somebody who has a fatty liver starting to develop, and there's an 86% certainty, an 86% suggestion of certainty that this is the case. This is fantastic from a clinical perspective. I mean, you took the bloods, you sent them out, came back, a report appeared in your inbox, and now you're looking at that, and you can sit down and think about, now you're already thinking about things that you can be giving your client and making interventions, and you haven't had to do that much to do it, other than to be what you're good at as being clinicians. When we look at the biosynthesis pathways, which I spent a minute or two going on one, if we now enter into the world of genetic mutations, single nucleotide polymorphisms. So in order to go to make purines and pyrimidines, we need a folate pathway which is operating appropriately. We need to be able to go down through dihydrofolate to methyl tetrahydrofolate without there being any SNPs or single nucleotide polymorphisms in the way. And as we go down, there's one particular enzyme here called SHMT1, which is responsible for this serine to glycine pathway. This is a seriously important pathway because the majority of glycine you find in the mitochondria. And dysfunctions in this particular little area here is also going to give you a hint towards mitochondrial dysfunction. Certainly people would be mitochondrially challenged with that kind of SNP there. And this requires cofactors, and this requires B6 as a cofactor. So now if you've got B6 issues, and we looked at the test and we can see that, let's say, for example, MCHC is low, we're thinking B6 is a problem. If we're thinking B6 is a problem, we can also then think further down over here at the homocysteine level. This is outside of nucleotides, of course, but we're now thinking about how we're going to get down from homocysteine through to glutathione, because B6 is required to convert this through an enzyme called CBS or crystalline beta synthase. But when that's down, that enzyme's not working. You're not going through the pathways to either purines or pyrimidines. And of course, down here at pyrimidine synthesis, we need magnesium as well. You need magnesium in the mitochondria, which I think is complex 5. So if you're challenged for magnesium, which surprisingly enough, most people seem to face huge challenges for magnesium in this day and age, let alone zinc, there are going to be problems there. I thought I'd put this in again just to put it in from a different perspective, because every time I see one of these pathway diagrams, it's either from left to right or right to left. It gets confusing, so I thought I'd put them both in here. But simply, here's your pathway into pyrimidines. Here's your pathway into purines. Purines lead to a uric acid. And you can see the cyclical nature of recycling that goes through. The human body is fantastic. It recycles. We're looking at the cofactors. We want to look at folate. So all of these markers can show you folate and B12, B6, iron, hemoglobin, MCV. All of those are factors you would take into account. And magnesium, you'd like to look at, obviously, magnesium. The red cell magnesium is the best form to assess. However, serum magnesium is a reasonable marker, along with albumin and calcium. The pathway, again, when it's all put together in a much more complicated way, when we go down into DNA synthesis, this is pyrimidines, we can see here this is the transport and circulation leading through to the storage in liver for the entire folate pathway. I know this is probably a bit more information than is required, but the idea I'm trying to put over to overcome this, nucleotides rock. Now, I was at a seminar recently on autism, and the person who was giving this, actually there were two doctors there talking about the same thing about folate, giving one doctor was talking about the need, how poor levels of folate were leading to elevated degrees of autism. And the other one was saying how he overcame that by going up to something like 20 milligrams to 50 milligrams of folate as a supplement. I haven't got the nerve to give those kind of dosages when, 400 micrograms is about as high as I like to go, maybe 800 in certain cases. But we're talking huge amounts of folate. And I thought about it afterwards, and I actually went up to one. I said to him, the fact is that you're leading to that because it's the DNA expression that's being compromised. And if you put in nucleotides into there, you're overcoming those folate pathways. The SNPs exist, but you don't have to go through that entire synthesis program. You're putting in readily available to be used immediately. It's like going into a shop and buying, I don't know, if I buy a suit off the peg rather than going to a tailor and having it made to measure. Just that's the sort of analogy. If we look at pyrimidines, we're going down this de novo pyrimidine biosynthesis pathway, and we go through the orotate and dihydroorotate pathway, and we come into the uracil, and here the salvage pathways kick in, and here the de novo synthesis pathways go in. Now, I'm not talking about the purines because purines I don't consider to be as much of a problem as pyrimidines. I think it's pyrimidines that are the biggest problem, and it is the pyrimidines in the formulas that are being used that are creating so much success. This is the orotate pathway. Dihydrotate orotate, DHO, this is an enzyme which catalyzes dihydroorotate to orotate. And that orotate then goes to become pyrimidines. So then pyrimidines then go to DNA and RNA. This happens to sit in the mitochondrial membrane, and this is important from an energy point of view, perspective, that this is where CoQ10 binds to. This pathway can be inhibited by nitric oxide. And since orotate is the precursor of all pyrimidine nucleotides, inhibition contributes to nitric oxide or NO- dependent cytotoxicity. So you can test for this. This is going to be important because this CoQ10 is essential in the formation of orotate. So it's a double. It happens to be there for a reason, that pathway. And if that is blocked, which I'm going to talk about, there may be significant issues when you think about statins. So I'm going to say, hypothesis that anyone on statins should be taking nucleotides because that is one of the pathways that's going to be blocked. So CoQ10, essential component in the synthesis of pyrimidines. The dihydroorotate dehydrogenase, that DOH2 whatever and they do, is the fourth enzyme along in the pyrimidine synthesis chain for activity. This pathway is blocked because of HMG-CoA reductase nature of the way that statins work. And if it's blocked, I say that statins therefore should be harmful to immune health, RNA and DNA production, lipid synthesis, red cell membranes, because pyrimidines are essential in the formation of lipids, and they're essential in the red cell formation, the membrane formation as well. So all the side effects that people get from statins, like muscle pain or whatever, I did this last night, so I'm just thinking I should consider the statin side effects and see if there's a link. So that this is an area for research. But I've not heard of this before, so this could be something novel and new and something we could do. Moving aside from blood chemistry for a moment, and we look at the Great Plains, which is an organic acid test. They happen to do an orotate or orotic acid marker. So you can assess orotate levels. We look at pyrimidine metabolites here. So we've got uracil and thymine as well. If thymine is extremely high, then you need to be concerned. This can be a precursor or a marker for cancer development because it's a folate level. I happen to think if I go back to that little formula that U plus CH3 equals T, in other words, uracil plus methylation equals thymine, we're now looking at possibility that it's down to uracil and the lack of methylation that's leading to elevated thymine. Lipid synthesis, essential in lipid synthesis, and now think of cell membranes. Nucleotides and cell membranes. As soon as you see there's a need for nucleotides and you find that there's an energy problem, think about cell membranes. There was a guy called Leslie Simpson who sadly died, I think it was early this year, who formulated a lifetime's work, unrecognised, about cell membranes and different red blood cell shapes causing as being causative factors in ME and chronic fatigue. And he said, and the marker you want to look at is ESR, erythrocyte sedimentation rate, and if it's between down from 2 to 0, there's a problem with the cell membrane. If there's a problem for the cell membrane, I think we might need to have pyrimidines put into the diet. So, why use something like FDX? Because you need to look at all the issues that might occur. For example, we've got megaloblastic anemia. Pyrimidines are essential for red cell formation. They're essential in the formation of the membranes. As I've mentioned, orotic acid is an intermediate product in pyrimidine synthesis. If there's a problem there, you're going to have end up with something, or potentially end up with a megaloblastic anemia because pyrimidine synthesis is down. This can also lead to autoimmune disease. There are links between rheumatoid arthritis as well. All of these factors, lipids that pyrimidines are essential for, that go into the red cell membrane, can lead to these problems. If your red cells aren't functioning properly, you're not going to get oxygen delivered to the cells. You're not going to get them delivered to the peripheral cells that require them through the capillaries. If somebody's capillaries are narrower than usual, which is a possibility, and you've got odd-shaped cells, they're not going to get through. You're going to have people who are energy challenged. They are going to go through the typical ME kind of symptoms of being able to function 1 minute and being flat out the next. This is a problem. When Leslie Simpson did his observations, he happened to notice, and I use this as an observational backup to what I've been saying, that the cells regain shape with B12, which makes me think that B12 and red cell formation is important. Nucleotides, again. ESR, as I've mentioned, and I suggest that if you have patients that are fatigued, then use nucleotides as a form of supplementation, as an essential form. Nucleotides lower the threshold for lower stress thresholds. They have a direct effect on cortisol build-up. I'll change my name to Spooner, I think. And essential in conditions of stress. Now, everybody is stressed out. Nucleotides work really well in the sports world. I know. I've been told about great recovery times. This is one of the key reasons when you ameliorate the effects of stress. Basically, single markers only show up on a test if they're outside of pathological ranges. So what should you do? Is it hard to join up the dots? Yes, you need lots of time to do that, and time is a precious commodity. I want to leave with this because I like this, because I want to explain what Functional DX is about. So I'm going to leave you with my little cartoon, which I'm very proud of. So I want you to meet the Norms from the mountain of Norm. This is Norm and Norma. And here they are, happy, healthy medical norms living here, because that's what we're all told we are, healthy medical norms. But Norm, like us all, worked hard, Little thin stick man, constant worry and stress led him to becoming tired, exhausted, and eventually filled out and gained weight. So he wasn't such a little thin stick man anymore, like me. So he went to his GP, and his GP said, nothing wrong with you. Your glucose levels are perfect. Your cholesterol levels are perfect. Everything is it should be, but it wasn't. So Norm ended up here, where people usually end up. And he found himself down from the mountain on the plains of Farmer, looking up at his mountain home through his hospital window. So there was only one thing to do. Escape. Because he missed his home and he wanted to go back, like anybody in hospital. But once you're down from the mountain, of Norm, and you're on the plains of Farmer, getting back up again to where you want to be is difficult. So the moral of this tale is don't let the norms of this world slide down the slippery slope. Catch them early before they slide down to the plains of Farmer. And that's where I'll leave it. Thank you.